Thank you, Dr. Amarek and Linhares, for the invitation to be here talking about implementing the use of whole genome sequencing for PRRS decision management. And this is a follow-up and part of the study that Daniel presented before, so make my life easier. So when you think about PRRS virus that we are talking, and if you take the DNA and stretch that out, we'll get basically 15,000 base pairs. And the image that you see in the screen is just a representation of that. But one specific region that is the OR5 is what we have been using for PRRS virus decision and management in the cell herds and finishing herds. So when you think about the usage of OR5 and PRRS virus whole genome, I compare that to an iceberg. The OR5 is the tip of the iceberg, and now that we have more tools and start to understand that is using whole genome sequence, we can dive in and better understand genetic structure and how can we take informed decision about that. So if you look for the OR5, it represents basically 4% of a genome. We use nowadays to classify the strains according to LFLP, lineages, and there is a common interpretation that and is distinct across systems. 1, 2, 3% different represents a new strain in the herd. On the other side, when you think about the whole genome, we still understanding and using this information to classify PRRS virus, we can get a better characteristic phenotypic of these strains. We can understand if there is genetic evolution, recombination, mutations occurring in the gene of this virus, and if multiple strains are present in the sample, we usually get genome fragments, and you can identify them if you have a farm reference strain. So this is a chart from our work that we did. We sequenced here more than 100 samples for PRRS virus, and we use it on your y-axis. You have CT values. The specimens is each of those bar that you have there. But one thing that we can see here is that CT values lower than 22 had better success rate for whole genome recovery. So if you are looking for recovery of whole genome of PRRS virus, target samples that has lower CT values is the most recommended for this approach. On the other side, if you look for the upper side of the bar, we don't have a good whole genome sequence recovery rate, that is those red dots over there. Well, but if you look at the big box, those are processing fluid samples, and is a population sample. So the other thing that we can infer here is that the sample type matters, in the way that if you're looking individual sample types like lung, serum, for the whole genome sequencing, they are more suitable to get a full genome recovery. On the other side, if you're looking for samples types that is aggregate sample as processing fluids, oral fluids, that you have many animals in the herd contributing for that, they may be more suitable to recover different strains virus that are there circulating in the herd. So each sample type may have a different approach to be used here. So Daniel already touched base on this, that using whole genome sequencing in this study, we found that herds that had up to 2 PRRS virus strains reached stability 12 weeks before the herds that had 3 or more PRRS virus strains detected in this time of after low exposure, and also that herds that has up to 2 PRRS virus strains had fewer piglets lost than herds that had more than 3 PRRS virus strains, and that was basically 800 piglets difference per 1,000 sows that were there present in the herd. The other thing that we look at, and is a hot topic, is recombination, and we always got scared about that when you say, oh, we have a recombinant strain. But from 70 farms we were able to get a whole genome, and comparing those with farm recombination in 12 of those 70. The reason that I say we got scared is because we don't understand well, but the reality is we have more recombinations occurring in the field than we think that they are occurring. And every time that we have those recombinations, we should just understand better how that implies our daily basis activities in the herd and not be scared. Because one other thing that we found here was that basically 60% of these farms, there was a recombination strain in farms that had more than 3 PRRS virus strains circulating in the herd. So the larger the viral load and variability that you have there, more opportunities for recombinations to occur in the, for this virus. And also we had 1,800 different in piglets lost per 1,000 sows than herds that did not have a recombination. That was a difference. And that may be because when the recombination occurs, you emerge a new PRRS strain, you need to go through a process of develop herd immunity to that PRRS virus strain and get some losses at those points. So if you think about how can you use next generation sequencing and understand that there is a willing to move from OR5 to next generation sequencing in this scenario, the first question that we need to ask is there a farm reference strain available for comparison? Well, if there is not, then you may not take action. You don't go for a whole genome sequence. You can keep using OR5 to do your analysis. But if you are thinking the next generation sequence is a tool to be used in your herd, go forward and recover a farm reference strain for a comparison. If you don't have that, your work gets a little bit complicated to be developed down the road. Well, now, if you got the whole genome sequence recovered and you have that as a farm reference strain, do we need to sample every week and submit our samples for next generation sequence? Maybe the answer is it depends. And you need to answer other questions. Example, is there increasing aborts, piglet mortality, dropping CT values on samples that you are using to monitor? And if your answer is no, maybe you don't need to go for whole genome sequencing. You can keep using the RF5, using clamp technologies to understand if there is that's a vaccine-like or is a wild-type strain that is circulating your herd. Now, if the answer is yes, there is clinical signs occurring in the herd, there is pig mortality, drop CTs, whatever, then you need to start understanding, well, our goal for doing whole genome sequencing is to understand if there is genetic evolution from that strain that we had in the break, then use individual samples like lung, serum, try to recover whole genome for the comparison with your farm reference strain, and then you start to see that picture evolve over time. Now, if your question, and that's not only this time, but other times is, well, is there presence of multiple virus strain in the herd and you want to detect that, then go for aggregate samples that can be processed fluid samples, oral fluid samples, familiar fluids, or maybe do a pool of many serums there. Because you are targeting to understand if there is more than one PRRS virus, you want to recover that. Well, if you do that and sequence this sample, you may not get the full genome, you may get the fragments of genome, and that's expected because if these strains are similar, the technique still need to evolve to recover the full genome. But when you compare farm reference strains, you can understand if there is more than one strain there circulating, and you don't need to recover vaccine strain because those sequences are publicly available and you can differentiate if this is a vaccine that you are using in the herd or if it's a second or a third wild type strain that's out there. So with that, I would like to acknowledge Iowa Pork Board and Pork Checkoff for providing funds for this study and all the veterinarians and producers that did enroll audits and walk with us through this process of understanding how to use next generation sequence to the project. And he's a picture of our team at Iowa State University, where we are proud of being part of this team and helping producers and veterinarians with information about PRRS management and control and other things. So with that, thank you very much for the opportunity.